So I will elaborate a little bit on how the field is, where the field is standing at the moment regarding metabolism, and show you some data regarding that, and also try to say something about hypotheses that's on the stage, and how things are connected. So there are not any objective hypotheses or theories that everybody agrees to, but there are some observations that fits very well into classic concepts, and that we can, I think, have quite good confidence about. And I have nothing to disclose. So we are working in kind of a translational setting where we get samples from the clinic, and our clinic is Haukeland University Hospital, and there is where doctors Dr. Flugen and Dr. Mella is located, who has done quite some clinical intervention studies in ME/CFS patients. So they have collected samples, and I came into this field about 8 years ago, and I'm a molecular biologist and cell biologist and do experiments in the lab, and we got funding to go deeper into investigations of these samples from the clinic. And we have tried to interpret this and also make hypotheses and also test some aspects in the laboratory. So these are the two major clinical trials where we got our samples, and these two alone provided nearly 200 or samples from 200 patients, and also with this comes quite a lot of information about the patients, so we have quite some data to link to the biological measurements that we do. So this has been instrumental for our work. So we have done metabolomics, measuring as many metabolites as we can do in those samples, and this is published. I will show you some data, and at the moment we are trying to dig into and write a manuscript about where we have measured proteins. So we have done quite deep protein profiling in the same samples, and when we have both these, we can start linking things and try to understand at a deeper level also. So this is Bergen, for those who have not been there. It's a beautiful small city in Norway, but I say small, but it's the second largest actually. So, but in international context, this is quite small. And this is the hospital, the university hospital, and our department is actually like in the middle here. You see the greenish building there. That's us. So we have actually interactions at the weekly level to try to coordinate activities and discuss results and hypotheses. So I will take you back to some basic concepts of energy metabolism in the cell, so cellular energy metabolism. So this is a cell, and here you have these mitochondria. We have already heard about those. That's organelles, so they are similar to our organs. They have specific functional roles inside the cells. We have heard that the mitochondria, that they are the power plants of the cell, but this is only one of the functions of the mitochondria. There have been, the last decades, there have been extensive research on all the different roles and aspects of mitochondria, and they are involved in more or less every disease. But in this context, their role is quite clear, I would think, clearly linked to the process of producing energy. And energy, when we're talking about energy production inside the cell, we're talking about ATP production. That's the main energy currency inside the cell. So typically what happens in this main main energy pathway under aerobic conditions is that glucose is taken into the cell, and then it's degraded to something called pyruvate, so it's cleaved to a 3-carbon unit. Glucose is a 6-carbon unit, and then it's taken into the mitochondria, and here there is an enzyme called pyruvate dehydrogenase. I will come back to that. And this cleaves off 1 carbon and makes acetyl-CoA, which is taken into this Krebs cycle or the citric acid cycle. And this is all dependent on the presence of oxygen. So when oxygen is present and there is enough glucose, quite some ATP is produced. Most of it from the mitochondrial production, but also some from these initial steps. So this is what provides the main source of ATP for the cell. And what Systrom talked about earlier today, it manifests in a way the ability to supply oxygen for keeping this process going. So under hypoxic conditions, there is a limitation in the functional activity at the mitochondrial level because there is not sufficient oxygen. And this actually just shut down the entire mitochondrial system, and the cell rely on the ATP coming from this process, and it has to compensate by degrading more glucose to get compensated more ATP. And this leads to an overproduction of pyruvate, and this has to be secreted, and it is converted to lactate and secreted as lactate. Lactate is not a kind of an toxic compound for the body. It actually has important roles, but it's a very clear manifestation that you are having these hypoxic conditions. But the cell cannot continue in this way for a long time. In the blood, we will of course see this as a rise in lactate, and we know, I think most of us, how it is to have this feeling of a rising lactate. It's not very pleasurous. It can be it can hurt for a normal person, and also the patients describe this feeling of lactate accumulation all the time. We hear it quite often. So we think this is very relevant, and also it's very relevant in context of the findings of all the people who have published research related to impairment of blood flow in the body. Now, we did a study which was published in 2016, where we measured some metabolites, amino acid metabolites in the blood, and we could interpret those data we find a pattern that we believe stems from an inhibition of the pyruvate dehydrogenase enzyme. So with this blockage here, you will get the same result. You will have an increase in lactate, and the mitochondrial oxidation machinery is turned down, and you have a pressure, and you have a reduced ATP production. So here you have more lactate, less energy, and also if this becomes something chronic, of course the cells will need to look for other energy sources than glucose, the glucose-driven pathway as well. So this will lead to quite many downstream effects in the body. And this is what we want to understand more about. And when we're doing that, we are doing all these measurements of metabolites and cofactors and different things that circulates in blood and that interacts with this system. And amino acids and fatty acids can fuel into the main energy pathway to compensate for parts of it, but this is most of it is dependent on a functional mitochondrial system. So this is quite complex to try to understand, but it's also we also know quite a lot about how the system respond to impairments at different levels. So we can try to do some interpretations, and if possible, we can also try to find ways to compensate or decompensate or improve certain aspects. And there has been some publications in the field, and probably as many of you already know quite some about this, and these are not all, but we think that the different metabolomic studies that has been done using different technologies and different cohorts maybe or probably is different flavors of or reflect different flavors of energy stress inside the cells. So this can be interpreted and you find patterns that indicate stress energy metabolism. And we also have different ways to look at those data and interpret them. I think they are compatible. It's just that we are focusing on different aspects and putting them together different ways to find ways to target them. But I think both organelle dysfunction and that primarily is mitochondrial dysfunction, as mentioned here earlier, can be relevant. The supply of fuels and oxygen is relevant. PDH inhibition I mentioned, and also the kunin pathway, that's the tryptophan metabolism, has been some hypothesis that has been circulating in the field. Also other hypotheses. But although we have quite some findings really showing that there is something happening here, we have not found a common metabolic phenotype for ME/CFS. And that is something that I'm not sure if we will ever do that, but I think we can find some common metabolic elements among patients. And that can tell us also something about core mechanisms and very important mechanisms underlying this. But because there are so many layers of adaptation, this is the most vitally important process in the body, in the cell, to keep energy fueling going. And the cells have evolved protecting all these mechanisms. So it can shift easily from one mechanism to another, one pathway to another. So it is very important to keep this going. And this also makes it quite complex and difficult to find exactly where there may be impairment. And this is an example of how it looks when ME patients do exercise training. This is from a study in Oslo, Katarina Lien. So this was a repeated CPET test where we have blood lactate on the y-axis, and here is the power output. That means the workload for the subject on the x-axis, and we can see the red lines here. That is far left compared to the blue lines, and the blue lines are the healthy controls, and the red lines are the patients. So clearly there are differences there. And super athlete, and actually I've seen Olympic champions that do the same. They will be to the far right here. And in addition, they will actually probably be able to accumulate even more lactate because they have optimized the machinery for tolerating it in the body. So there are many factors that can come into play here. We heard from Systram earlier today that these kind of effects related to blood supply is probably not only driven by deconditioning, and we strongly believe that. We believe that the patients have an abnormally low workload threshold before the accumulation of lactate starts. But there are ranges of normal activity or normal differences there. So we need additional data to really understand it. When we look at baseline levels, here it seems like the patients maybe have a little higher lactate compared to controls. But when we have looked at this in our studies, we have not seen that being documented that at baseline or at rest does not seem to be a higher lactate level, but we have observed single patients and measured lactate, and we can see that even just a small excess, a small walk can lead to a significant raise in lactate. So it is definitely something that could be probably used as a better marker also if done systematically. So we have been looking into different metabolic mechanisms that comes into play at the cellular level here, and there we can utilize tools from other fields of research like cancer and neurodegeneration and diabetes experiments, because there you have tools to investigate the metabolic programs inside the cells, and we can mimic parts of this and ask specific questions. And in one example, we took information from this 2016 study and from the PDH inhibition, and we cultured. This was normal human muscle cells we cultured, and we exposed them to blood serum from ME patients, and we could see that this alone led to an increase in lactate production and an increase also actually in oxygen consumption. So we used this seahorse instrument to do this. Then you can mimic a maximum oxygen uptake test in the dish, and you can go into detail of which part of the mitochondrial system that is not working properly. So this shows that there are some factors in patient blood that affect cellular metabolism and lead to something that we think looks like a stress response, an energy stress response. We also took another piece of information. We could measure an increase in an expression of a protein called PDH kinase 4. And we took that information and we overexpressed this factor inside culture cells. So we put in extra genes for this. And this is a factor that actually inhibits the PDH enzyme. So by doing this, we could see that the cells shifted from this glucose degradation to fatty acid degradation as an energy pathway. It's not, it is a known or anticipated effect of inhibiting PDH, but it clearly shows that you can take mechanistic elements from the patients and you can mimic them in culture, and then you can start doing more detailed experiments to find out how it all is related. So these curves here, they are from the textbook in biochemistry for the medical students, and shows what happens with energy fuels during a period of starvation. So 4 days, 4 days, 6 days, 8 days. So the underlying curves there are normal. First you see a drop in blood glucose, but then it stabilizes because the body is optimized to stabilize glucose and to not fall too low. But then the fatty acids come into play and it increases, and that's the non-esterified fatty acids, free fatty acids. And at around day 2 or 3, we really see the ketone bodies coming up. They are produced from fatty acids, and they are a reserve fuel for the brain, because brain is dependent on glucose, or if not glucose is present, then you can go for ketones. And when we do measurement, and we have seen this repeatedly, the ME patient seems to have something that lies, is a kind of an intermediate of this, like a baseline. So there seems to be an elevated energy stress situation chronically. So we believe that this pattern reflects that there is an underlying energy limitation that can contribute probably both to symptoms and symptom worsening if it's triggered even more by increased energy demand. And we did this global metabolomic study where we tried to go also into see if there can be subtypes of ME patients that express different phenotypes, that they are different in some way. So we got quite a lot of data from this. We measured first around 600 metabolites and tried to see what happened with those. And we do quite advanced bioinformatics, and these two first figures here are simply asking the computer to group or cluster patients that are similar to each other. And then we found 3 subtypes that looked quite different. We can see it here. Here are the controls, the yellow ones, and here you have one subtype of ME, and here you have one subtype, and then you have a third subtype that look more similar or in between the 3 others. So this, one additional aspect that made this quite interesting was that we could see between the 2 major metabolic subtypes, we could see a difference in function scores. So one of the subtype, the metabotype 2 we called it, had a worse function. So this tells us that there seems to be an association between the metabolic phenotype, how metabolism is regulated, and how severe the function is affected. And we don't know, we cannot say if this is a cause or a consequence. We can only say that there is an association. But of course, lying in bed and if even having an effect on the functional activity level can affect many things in the body. So it is difficult to say. We also looked for different other confounders or factors that could influence those data. Of course, we know about sex and diet and drugs and age. Many things affects metabolism in the body. But when we did this subgrouping of patients, we tried to use robust methods so these elements would not contribute to the subgrouping of the patients, and we tested this. So we feel quite confident that this clustering patterns was driven by ME itself and not bystander effects. This we think the data reflect both contextual adaptations in the patients to an underlying energy stress, but also we think there are elements that are common. We did find common or more homogeneous changes also that can maybe be related to more underlying mechanistic processes. Yeah, and we also validated some of the findings in bigger cohorts and looked also at some metabolic stress hormones that supported the biological relevance of these subgroupings. So there is a struggle for energy, and we think that these are connected to the decreased oxygen extraction and delivery in the body, or the lactate overproduction, and that this can contribute to physical debilitation and also contribute to make it worse or better. And it links quite well with other findings in the field. Just at the end, I have a couple of slides that takes this a bit further, because it may not only be what happens, the acute state. It can also be that activity can lead to release of, call it, damaging factors or accentuating factors that makes symptoms worse. And this is, of course, relevant when talking about post-exertional malaise. What factors can be linked to to the mechanisms of that. We think that there are maybe mechanisms at the cellular level that contribute to post-exertion malaise. Maybe it's something related to the waves of regulation occurring during normal training or excessive training. And we also have to look into different periods after exercise to find that, and Maureen Hansen and other groups have started that journey already. So I think this is something we need to focus more on in the years to come. Our main hypothesis, how this is linked together, is that we think there is an underlying autoimmune mechanism of some nature. I think this is a leading hypothesis in the field. But we also think that this autoimmune mechanism is quite closely linked to the vascular dysregulation, and that this leads to different kinds of adaptations and symptoms in the patients. And by this I want to thank my collaborators in Bergen and also outside Bergen, we have both collaborators that's contributed to the clinical intervention studies, with the Fluge and Mella leading those, and we have people in the lab and also collaborators that contribute in clinical trials and also in more specific studies on and analysis. So thank you very much.
Thank you, Karl. Questions? On the front, Richard.
Hi, I just wanted to let you know that there's actually a patient-led study going on right now about lactate levels. There's patients organizing, I'm one of them, to measure lactate levels at home with the home devices. And Todd Davenport actually put together an official survey for us through his university. And people with ME and long COVID are finding these high lactate levels at rest, and then measuring after breakfast, measuring during PEM. And so we just put the survey out about 2 weeks ago, I think, maybe 1 week, and I already have about 150 inputs. And so hoping, by the end of June, we'll have a lot of data to actually analyze that.
That sounds very, very interesting, because I think also that probably it's better able to reflect the true condition of when they can measure it at home under their own, can do it themselves. Of course, that is much less exaggerating than these samples has been. So very interesting. Yeah.
One patient actually measured it 17 times during a day. So he showed that I think it was just 4 or 5 of them had extremely high levels, and the rest of them were more normal. They were still elevated a bit. So I definitely think it's interesting to be able to do it at home in different conditions.
Very interesting. Thank you.
Any thoughts on the ketogenic diet?
Maybe. The ketogenic diet, that will be to try to circumvent some of the limitations in this type of metabolic inhibition, and is used if there are certain mitochondrial dysfunctions and also different conditions where this can have a beneficial effect. We only hear about some patients that have tried it, and reports are both ways. Some have positive effects, some do not have positive effects. Some true, somebody try something in the middle, just reducing calories or carbohydrates. As far as I know, the keto diet has not been tested in clinical trials yet. So I guess that's if we want to answer that question properly, we should do it. Yeah, but maybe there are also other ways we can try to compensate. Yeah.
Any last question?
Thinking about standard blood tests for things like lipids and the levels that doctors apply, is it perhaps the case that the altered metabolism means that they should be applying different criteria or different reference levels? Have you got any insights into that? I say lipids, but there could be other things that comprise standard blood panels that might be out of whack. Got any thoughts on that?
Yeah, I could try. So these differences we have seen regarding lipids and amino acids and glucose, these are within the normal range. They're within the normal biological spectrum in a way. So the body are using these metabolic shifts to compensate something. And we have not patients that have diabetes or something like that. But that said, I think it would be possible if you use some other reference levels that you can say something of which metabolic phenotype one patient seems to be similar to, and maybe then you can start, a little bit more about the metabolic limitation of that patient. I'm not sure if you can do something very practically to help that patient, but you can at least think about diet and what's possible. So there are no tests established. We have not. I only know how these patients and the subgroups look like, and it seems that 1 or 2 measurements should be enough for saying, okay, they look similar to this or that. But I'm not sure if this is done. Is standardized for most of the patients. We did not, for instance, take the samples here standardized under fasting conditions. So that here there are a baseline variation from the start. And of course, then you get limitation related to insulin-regulated processes, for instance. But still, if you do a measurement with a quite large group and you see a difference at group level, you think there is something that tells something. And some of the processes we have seen are insulin dependent, but some are not. So, yeah.
Excellent. I didn't see any more raised hands, so we're pretty much on time. Just to thank again Karl and Dag for their presentations. Thank you.